three processes of information: replication, transcription, and translation.
A DNA contains the complete genetic information that defines the structure and function of an organism. So, the proteins that are forming here are being encoded information by the RNA. It is very essential in a protein. Thus, within most cells, the genetic information flows from DNA to RNA and to protein. So, this is my setup for the three processes of information. First is for the DNA. We can see in this setup that the side part of the DNA is the sugar phosphate. Peach color represents the guanine. Yellow-green color represents the cytosine. And the blue color is thymine. Green color is adenine. For the three process, first is the replication. In replication, a new DNA strand is being duplicated. In here, we can see that in this side there is a daughter DNA, and in this side there is a daughter DNA. So DNA is duplicated into two. The double-stranded nucleic acid is duplicated to give identical copies. So we can see it here. Then for the transcription, a DNA segment, this will turn out to be a one-stranded RNA. It is a single-stranded sequence of RNA. Then the RNA moves to the nucleus up to the cytoplasm. And for the translation is the RNA sequence. From the transcription, it goes to the translation. The RNA is being translated into an amino acid. And we can see that the first three codon is being translated as the arginine. And the second three codon is being translated as the leucine. And the third three codons is being translated into arginine as well. Then the last three codon is the stop. So in the bigger picture, the central dogma of molecular biology is an explanation of the flow of genetic information. And it is being first stated by Francis Crick in 1958 as once information has passed into protein, it cannot get out again. In more detail, the transfer of information from nucleic acid to nucleic acid or from nucleic acid to protein is possible. But if the transfer from protein to protein or from nucleic acid to protein is impossible. So what really is central dogma? Central dogma is a process in which the instruction in DNA are converted into a functional product. It suggests that DNA contains the information needed to make all of our proteins, and the RNA is a messenger that carries this information to the ribosome.